this the same species across the world. And genetics tells us, and other studies talking about genetic diversity, really imply that this is very unlikely that this is the same species. So if those are the places they were described from, these are the places that I have samples from. So we have it all the way from over in Yemen, we have it from Australia, Indonesia, all across Micronesia, Japan, and Hawaii. So if you've never seen a phylogenetic tree before, it's not as scary as it looks. We have up here different clades, what we call clades. And the closer each clade is to each other, the more likely they are to be related to each other. So over here, this is made from, what did I say? So 180 specimens and 65 haplotypes, so 65 independent sequences. And what you find, I'm just going to point out a couple really interesting things. So Guam specimens, you can see up here Guam, and then down here as well. Guam kind of pops up all over the place. And one thing that's really interesting for this study is that the West Coast specimens of Guam are actually more closely related to those in Japan than they are to the specimens from the other side of Guam. I think that's really cool. And then on top of that, you have a lot of connectivity between different islands in Micronesia. You have Pohnpei and Chuuk have a lot of connectivity, so there's a lot of sharing of genetic material, and between Pohnpei and Kazurai, but nowhere in here did I really see any place where Chuuk and Kazurai kind of mix at all. And that's really also interesting too, and probably has to do with some ocean currents. And currently in the literature, there's a really popular way of species delimitation, of determining what a species is. And again, if you like the technical details of that, you can talk to me afterwards. But so using these two programs that are very widely accepted among many taxonomists, the species delimitation says that in this clade alone, there could be 35 to 37 independent species, completely distinct from each other. Whereas, like I showed a little while ago, we were thought that there were only 4. And this is a similar tree, but from a different gene. And it's pretty much showing the same thing. The topology is very similar. The only thing that we do have up here, this is Actinitrichia taiwanica, and this is the one that it was actually described from over in Taiwan, hence the name. But it is so distinct, it almost looks like it could be a separate genus. And this is something that no one has noticed before because no one has actually gone and taken this many specimens and examined it on this level. I gotta wrap it up so quick. So basically what I was gonna say is that morphological diversity in algae is really difficult to classify. The pieces of it that you actually look at are very difficult to tell apart. So if you were to look at this little this chart here, you have a massive overlap between what they say each species is. So here are the 4 species, and like the branch width, 400, 400 to 650, 400 to 600. So all these published studies are basically publishing and saying all these things look almost identical, but they're not going to say that because then they wouldn't have a study to put out. So my point being that morphological diversity makes it really difficult to do these kind of studies without genetics. These are some data I collected from my specimens. Same thing. You look at the width of the individual specimens that you're looking at based on the clade, and there's so much overlap that you can't distinguish between them based on morphology. Again, you need genetics to do this taxonomy. I think the only wild card that we have left in the mess of this wonderful project is that there's a species that was described from North Vietnam during the Vietnam War. And I love taxonomy because it is essentially a history lesson at the same time. And this was described during the Vietnam War. And it wasn't described very well as far as taxonomy goes. This is not a very good picture, not a very good study. And so until we can actually find a specimen from that area and get genetic material from it, it's difficult to actually make any direct distinctions as to what our species are. But this has a really fun story behind it too. So anyway, in conclusion, I know that was kind of quick, but so the diversity of Actinitrichia is a lot greater than we ever thought before. And the implications for this are quite, pretty big. If you're looking at conservation practices, and you're wondering about niches, and you're wondering about how ecosystems change over time, you have to have a baseline understanding of what it is actually out there. And it's, like I said, genetics is the best way to do that, not just for algae, but for many different organisms. Morphological characters are not useful in determining the IG. And no spoiler alert here for my thesis, but I will be describing probably at least 3, maybe more species for Guam and the Marianas Islands, and perhaps Australia as well. But you'll have to stay tuned for that one. Anyway, well, thank you, UOT. Definitely, thank you, Sea Grant, for funding most of this project, Dr. Days. And then also, Western Pacific Coral Reef Institute actually paid for our expedition to get samples from across the Marianas to be able to check out and see what all this diversity entails. And the rest of those folks, too. All right, thanks so much. I think I got to get off the stage.
Thank you, Anna. Are there any questions we're going to direct